Hello everyone. In this video we will be talking about solving combined spectra problems. And combined spectra problems are essentially where I'm given a variety of characterization information and I'm going to utilize that to solve for the structure of an unknown. And the information that we're provided is the molecular formula, a copy of the IR spectrum, and a copy of the proton NMR spectrum. And so from all of this information, I can piece together ultimately various different portions of my molecule, put that all together, and determine the overall structure of my unknown. In this video, we're going to talk about some steps on how to approach these types of problems, and then we're actually going to work through an example. So hopefully at the end of this video, we'll feel more comfortable solving combined spectra problems. So here are the steps that I take when approaching a combined spectra problem. So the first thing I take a look at is the molecular formula, and I solve the HDI. And again, the HDI tells me the number of pi bonds and or the number of rings that exist in my molecule. While this doesn't provide me with a lot of information, it can limit the overall number of possibilities, especially if I have a large molecule. Next, take a look at the IR spectrum and identify the various different types of bonds that exist. And from those types of bonds, I can piece together ultimately functional groups that could exist within the molecule. Then lastly, I'm going to take a look at the NMR spectrum. First, identifying the overall number of signals and identifying whether my entire molecule has a plane of symmetry or if there's certain points of symmetry that exist within my molecule. Then going to go and analyze each individual signal, taking a look at the chemical shift, the integration, and the multiplicity. Again, the multiplicity is important here to find essentially the connectivity of my various different atoms. From this analysis, I can generate what are called partial structures. So I can look at sort of small segments of my unknown. And then if I do this partial structure analysis correctly, essentially what will happen is if I piece together those partial structures, I can solve for the overall structure of my unknown. So now let's walk through an example. So in this example, I have the molecular formula C4H8O as my unknown. So the first thing I'm going to do is solve the HDI. So if I put that into my calculation, I can determine ultimately that I have an HDI of 1. So in my molecule, I either have a pi bond or a ring that exists. Next, I take a look at my IR spectrum. And the first thing I notice when taking a look at this IR spectrum is this strong signal at 1740. So this corresponds to the double bond region. And again, this strong signal is going to correspond to a carbon-oxygen double bond. So that essentially takes care of my one HDI. Again, there's one pi bond here, and so there's no degrees of unsaturation remaining. Next, I'm going to take a look at this 2973 peak, noticing that is below 3000. So this is in the hydrogen region, and specifically if we're below 3000, typically it corresponds to a carbon sp3 hybridized CH stretch. Then lastly, I'm going to take a look at these peaks here at 2763 and 2709. Notice that I grouped them together here in my table. So these signals are a little bit low to correspond to a carbon sp3 CH stretch. And since there's also 2 of them and it has this sort of characteristic sort of W associated with it, this is what we see with that aldehyde. So that CH corresponding to an aldehyde. So essentially from my analysis of my IR spectrum, I know that I have an aldehyde that exists in my molecule. Lastly, we're going to take a look at the NMR spectrum. So notice that I have 3 overall signals in my NMR. I have the peak here at 9.57. I have this more complicated splitting pattern here at 2.39. And then I have this larger peak here at 1.06. So also, when trying to identify if there are planes of symmetry, we notice what we want to do is add up the total number of integrations. So notice that I have, if I were to add up all these numbers, it equals 8, and that's 8 overall hydrogens in my molecule. So I don't necessarily have any sort of plane of symmetry that exists within this structure. So let's go ahead and start actually analyzing each individual peak. So I'm going to begin with this peak over here because it's kind of a unique region of the NMR spectrum. There's not really much else that can occur in this region other than an aldehyde. And it has an integration of 1. So it is a singlet. Again, the shape of the peak, there's only one peak. And so I can draw the partial structure of this aldehyde. When I'm drawing my partial structures, what I want to do is account for the integration and the multiplicity and the chemical environment. So again, it's an aldehyde hydrogen. And ultimately, that's what the partial structure is going to look like. This squiggly line here basically says that there's another part of the molecule over here, but in order from essentially my analysis of the signal, I can just essentially determine this piece of my molecule. Next, I'm going to take a look at this peak here, so this large peak here. We can identify that it is a doublet. Notice that there's only 2 peaks here in the shape of that signal. And another thing that we notice is that it has an integration of 6. So if I divide 6 by 2, that's 3. And ultimately, if we think about things in factors of 3, essentially we can think about that as 2 CH3 peaks that are in the same chemical environment. And if those 2 CH3 peaks are in the same chemical environment and they are a doublet, it's only going to see 1 hydrogen. So if I put that together, again, it's a doublet, my partial structure is going to look like this. So all of the hydrogens that I have indicated here are in the same chemical environment. So that's our integration of 6. They only see 1 hydrogen, and that's where I get the multiplicity of that doublet. Lastly, I'm going to take a look at this peak here, first identifying the multiplicity. So there's 1, 2, 3, 4, 5, 6, 7. So we would call that a septet. And so this ultimately, it's going to see 6 peaks, or 6 hydrogens plus 1. That gets me my septet. So that is going to actually correspond to the hydrogen that exists on this carbon here. So if I were to draw the partial structure, again, indicating this is the hydrogens, they're split by the hydrogens that aren't indicated, but we can see here. And then there are no hydrogens adjacent here. So there's not another set of hydrogens that are making this multiplicity even more complicated. So from these partial structures, again, I can kind of piece together these 2 things here. And I notice that I have this area of no hydrogens. That's going to correspond to my aldehyde. So again, if I put all of these pieces together, my final structure looks like this.